Hi users, welcome to my channel Learning with Khatak. No one can deny the role of artificial intelligence and machine learnings they played in the real world applications today. A lot of applications we are seeing in the real life based on artificial intelligence and machine learning, and obviously we know that data science is a backbone of artificial intelligence and machine learning. Why? Because we can perform different types of analysis. Actually we prepare our data to feed to the different models of machine learning, of artificial intelligence. So data science plays an important role while preparing your data. As we seen in our last video lecture, part 1, heart disease predictions, that's how we can prepare our data. Actually what we have did in our last lectures, that how to import our data sets, how to perform a univariate analysis. So in today lecture, this is the part 2 of heart disease predictions. One request: if you did not subscribe the channel, please subscribe the channel and also share with your friends, with your social circles. And before moving to the part 2, if you did not hear or listens the part 1, please listens the part 1. Do your practicals by importing the datasets. You can easily find the datasets of heart disease that is publicly available on Kaggle platform. From there you can download these datasets, and then I performed several operations in the part 1 video lectures. From there you can perform different operations on the datasets, and today lectures we will perform the bivariate analysis. In the first part, what we have did so far, if we have a look at the part first, we simply import our essential libraries, and then we import the data sets, and then we start the exploratory data analysis. Keep in mind, exploratory data analysis consists of several parts. In this part, actually we are going to prepare our data sets to feed into our model. So what we have did so far in the part first, that we check shape of the data sets, that how many samples or how many records are there and how many features are there. Preview of the data sets, we preview some records of the data sets, summary of the data sets, and data set descriptions, data types of or columns, important points about data sets. And today we also look at the statistical properties of the data sets, and then we performs the univariate analysis. Obviously in univariate analysis or features of interest is a target variables and findings of the univariate analysis. So important thing is what we have finds from the univariate analysis, or what are the findings of the univariate analysis. In univariate analysis we have covered that our feature variable of interest is target. It or features variables of interest is target. It refers to the presence of heart disease in the patients. Target is a categorical values 1 or 0. It is integer valued, as it contains two integers 0 and 1. 0 stand for absence of heart disease and 1 for presence of heart disease. If you look at this, uh, yes, look at this. These are our 0 and 1. 1 for heart disease and 0 for have no disease. 1 stand for presence of heart disease. So there are, if you look at this, 165 patients suffering from heart disease. Similarly, 0 stand for absence of heart disease. So there are 168 patients who don't have any heart disease, and then if you look at this, yes, the frequency distribution, look at this now. Out of 96 females, 72 have heart disease and 24 don't have any heart disease. Similarly, out of 207 males, if you look at this, 114 and 93, there are 207 male patient records in all data sets, and this 93 have heart disease and 114 don't have heart disease. So These are the findings of the univariate analysis, where we look the features of 6 with respect to target variable, with respect to target, that how many females have suffering from the heart disease and how many males are suffering from the heart disease. Now these are the graphical representations to show that how many male and females are suffering from the heart disease. Okay. Now description, the statistical data that we want to know. Statistical data plays important role uh, to know about the features, what features are correlated with the target variables, what features affecting mostly or positively the target variables, and what features are negatively correlated with the target variables. So if I run this hot dot describe function, if I run this code, look at this code, this show me these are my variables, features, age, 6, CP, and etc. If I move the record up to last one, so if you look at this, these are the different variables, my target, etc. Okay. Now the statistical features are, what are the statistical features? Count, total number of counts or total number of records of age 33, because there are no null values. We will look at the null values and how to remove the null values if there is any null values. That is the mean of age is 64, 60, CP, etc. Standard deviation, minimum value 25 percentile, 50 percentile, 75 percentile, and maximum. In multivariate analysis, where we will perform the correlations of different features with the target, we will look at these statistical descriptions later on. So these are the description that what are the mean of age with respect to target variables. So target variables mean is 0.54 and the mean of age is 4 point something. Okay, so these are the statistical descriptions, and then we want to know the data type of columns. How to get this dev.head info? If I run this code, simply look at this, that range index 303, entry 0 to 303, data columns, these are my data columns. And this is very important to note that is any columns have any null values? If yes, keep in mind we have to remove these null values from the dataset, otherwise our resultant classifier will not perform properly. So if we look at this, three important information are there: columns, not null, data type. So that is age 33 entries, there is no null values. If you look at the all features have no null values. Luckily there is no null values in the or data sets, and this is the type of data sets. If we look at the age and 64, how is it possible? Age means age obviously is integer. uh, uh, Similarly, 6 is also integer data types. How is it possible? 6 male or females? If we look at our data sets, we have okay. If you look at this, let me show the data sets here. Okay. Uh, okay, let me show here the data sets. Df hot dot head 1, that how which type of values in 6, 1 or 0. So since 1 or 0 are integer value, that's why the type of 6 is integer. Obviously you can explore the data sets when you download, then you look at the values, from values compare these data type with these values, you will simply come why the data types is associated, why the integer data type is associated with the age and 6 and so on. So These are my columns. From the column description, I just came to know that there is no null values in any columns and the data types of my columns. Okay, give more description to the columns. How what type of descriptions I can provide to my columns? If simply, what type of descriptions, what type of information I can get? Look at, if I run this code, look at this. Age means age, sex, 1 for male and 0 for females. CP, chest pain type, there are four type of chest pain: one typical angina, two atypical angina, three non-anginical pain, four asymptotic. So these are important while we when we were predicting, or in case of multivariate analysis, these are important to note about that. That 6 have two values, 1 and 0. If you look at this, 1 and 0 are integer. That why the data type of 6 features is that is integer. And how we perform this: info, age 1, male 0, and etc. So these are the basics information, are some extra informations we get from there. And then class label or target labels. What are the class labels? What are the class labels? If I want to perform operations df hot and then target and then dot describe, if I run this code, okay, there is some problem. Okay, dot describe. Okay, if you look at this, the target variables, total values of target variable is 303. This is the mean, standard deviation, minimum value, percentile, 25 percentile, and so on. If you look at this, from this descriptions, we will see all the descriptions about the data, like average, length, width, minimum value, maximum value, and so on. So we have complete our basics operations on data sets. That is, we just came to know about the data types of the columns. Is there any null values? What is the standard deviations? What is the means? What is the 25 percentiles, maximum and minimum values of the target variables, and different features? What are actually values inside the features? For examples, we look there, that's sex means 0 and 1, CP means 4 type of CPs are there, 1, 2, 3, and 4, and data sets, these different types of CP are represented in form of 1, 0, and so on. So now here in this case, from there we are going to perform our bivariate analysis. Bivariate analysis, that estimate correlation coefficients. What is the correlation coefficients? We will look at this, and then checking correlation between columns. Different columns are correlated with each others. How they are correlated? What are the correlation coefficient if we compare to features? And then correlation between features versus targets. That how other features are correlated with the target variables. If we run simply this code, what we have did, the correlation coefficient range from -1 to 0. Simply let me run this first code. This is the correlation. Target 1, obviously our important feature is target, and we want to see that how the rest of the features are correlated with the target. How much CP is correlated with the target, how much thalage is correlated with the target. The correlation coefficient range from -1 to 1. If you look at these values, all these values have in a range of -1 to 1. If we look at these values, CP have a correlation coefficient is 0.43. It means CP is positively related, maximally related, correlated with the target variables. CP and thalage are positively correlated with the target, and slope is vaguely positively correlated with the target. How? Thalwich is a correlation coefficient of 0.42 and slope is 0.34. If you look at these three values, maximum values is 1. So CP has a correlated coefficient of 0.43 with the target variable once. Means this is positively correlated with the target variables. Thalage is positively correlated with the positive. Acha, oh, if you look at this, FBS is negatively correlated with target variable, the target variables. What does it mean? That the FBS have no strong relation with the target variables. Similarly, if we look at the CA, majority of features are negatively correlated with the target variables. So all features of interest or these three, they have positively relation with the with the target variable. Okay. Now, analysis of the features positively correlated with the target. Now we want to analyze what features are positively correlated and what we can extract from these analysis. So first is obviously CP is mostly correlated with the okay. CP is mostly correlated with the target. Now we first performs analysis of CP, that is unique value, and CP. So how many values are there in CPs and what are the unique values? There are four values. Obviously, we have noted from the correlations, if the description of the column, where CP have four value 1, 2, 3, and 4. Okay, and now frequency distribution. If I want to know the frequency distribution of CP, CPs has unique values 4, 0, 1, 2, 3, and 4. How many records are associated with 1? How many records are associated with 2, and so on. So if you look at this frequency distribution, so CP has basically four values 0, 1, 2, and 3. 143 records are associated with 0, 87 records are associated with 2, and 50 patients are associated with 1, and 23 are associated with no. So this is the frequency distribution of CP. Similarly, if you want to count again the same data, 143. Oh, okay, this is the same data basically, that how many patients are associated with 0 and how many patients are associated with 1. Now if you want to visualize this record with respect to target, if I run this code, if you look at this, this is the CP. CP has four values 0, 1, 2, and 3s, and this is the target variable 1 for 0. So what does it mean? How we can interpret this graph? This graph interpret if a patient have acha, okay, let me show one thing before discussing this graph from here. Okay, CP means chest pain type. What type of pain the patients have? One, typical angina. Two, typical atypical angina. Three, non-anginical pain. Four, asymptotic. So means again, keep in mind these four types: one, typical angina; two, atypical angina. So if I look at the graph now from there, 0 means typical angina, 1 means atypical angina. So if the patients have typical angina, the possibility is that the patients having greater typical angina predict show from this graph show that patients have typical anginas are more risky to the heart disease. Similarly, if a patient have chain pest type of 3, are very less risky to the heart patients. So this is the analysis of CP with respect to target variables, that if CP value is 1, how many patients have the heart problem? 140. If the patients have chest problem once, how many patients have the heart disease problem? Similarly, if the chest patients have CP2, how many problems have uh, the heart disease, and so on. Okay. Similarly, if you look at this value, okay, let me show you one thing more. Uh, this is the group values. What does it means actually? Now this is more clear forms. Here in this case, if you look at CP values, CP values are CP variables contains 4 integer values 0, 1, 2 and 3. Target variables contains 2 integer values 0 and 1s. So it means for each CP values there are 2 target variables, that is 0 or 1s. Obviously, similarly for 1, 1 and 0. 1 presence of heart disease, 0 absence of heart disease. So the above analysis gives target variable values categorized into presence and absence of heart disease and grouped by CP variables. And this is the visualizations, that if CP is 1, heart disease rate is that. If CP is 0, heart disease rate is that. If CP is 1, heart disease rate is that. If 2, and so on. So this is the analysis of CP features with respect to target. You can also perform analysis for exchange, old peak and Thallich variables. How you can perform these analysis? If you look at this, uh, just writing here Thallich or writing any features name, you will get the respective record related, correlated with the target variables. Okay. So in bivariate analysis, we have performs that how different features are correlated with the target variables. Okay, let me show one thing more important here from this. Okay, this is the okay counts, CP counts and CP unique values. Okay, this is okay. Okay, this is the findings. Okay, now findings of the bivariate analysis. Here bivariate analysis is completed. In bivariate analysis, basically we have performed that how features is correlated with the target variables. For example, in case of CP, we can see that values of target variables are plotted with respect to CP in the above graph. Target variables contains 2 integer values 1 and 0 and so on. Okay, the findings of the bivariate analysis. Before moving to the multivariate analysis, just look at the findings of the bivariate analysis. Bivariate findings of bivariate analysis from the above two graphs are: there is no variable which has strong positive correlated. If we look at the record of this, these are the target variables and these are the other important feature of the data sets. These are the dependent and these are the independent variables. So if we look at the values, there is no variable which has strong positive correlation with target variables. For examples, if we look at the CP, has 4.43 coefficient metric coefficient correlation coefficients have the target variables. If you look at all the features, no features have a value zero point 0.5, have a correlations with the target variables. There is no variable which has strong negative correlation with target variables. And the reverse form, no variables have strongly positive correlated with the target. Similarly, no variables have strong negative correlated values. There is no correlation between target and FBS. If we look at the value of FBS, minus 0.02. So this is the lowest value. So FBS has no correlations with the target variable. The CP and thalage variables are mildly positive correlated with the CP and thalage has mildly positive correlated with the target variable. We can see the thalage variable is slightly negatively skewed. If we look at the thalage, 0.42174441, the people suffering from heart disease the people suffering from heart disease target 1s have relatively higher rate thalage as compared to people who are not suffering from heart disease target 0. The people suffering from heart disease target 1 have relatively higher heart rate thalage as compared to people who are not suffering from the heart disease. You can analyze this by simply visualizing the thalage variables, the thalage features with respect to target, as we have seen in case of CP. Okay. Now, multivariate analysis. In multivariate analysis, what we will perform in multivariate analysis, that is we will practically build a heat map to look at the all features with respect to the target variables. So this is the end part of part 2. In part 3, hopefully we will look at the multivariate variables and then train test split and then how to create our model and so on. So wait for the third part with the multivariate analysis and some other interesting analysis and generating or inputs, feeding to the models, predicting by the models, and so on. So thank you very much.